Welcome everyone. It's Friday, but Science Festival and National Science Week are not over yet. My name is Katrina Nguyen-Robertson, and I'm very excited today to learn about a female grasshopper that don't need no man in this masterclass delivered by Professor Michael Carney, who pointed out that I wouldn't need a double-barreled surname if we also didn't need men to reproduce. This is the last of 5 masterclasses as part of Science Festival that's showcasing brilliant University of Melbourne scientists and their groundbreaking science and discoveries. And just so that you're aware, because we're covering such amazing work, we don't want people to miss out, and we want to make sure that you can see this recording again if you want to. So we will be recording today's Zoom session. Before I begin, I would like to acknowledge the traditional owners of the lands to which we're all gathered. I'm streaming from the lands of the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past, present, and emerging. And I invite you to pop into the chat where you're coming from. I see some familiar names, but yeah, let us know where you are and whose lands you're on. I think it's important that we acknowledge and remember that First Nations people were the first scientists of this land and the first ecologists of this land, as they endeavored to understand the land, the sky, and the sea, and they passed down knowledge for tens of thousands of years from generation to generation. So we have a lot to learn, I think, about ecology. So how can a grasshopper thrive and be just as ecologically successful if it's breeding without sex? The matchstick grasshopper has evolved into an all-female species that doesn't reproduce through sex, and Professor Michael Carney will share the fascinating insights into that and what that tells us about evolution. So as a little bit of an introduction, Michael is a physiological ecologist in the School of Biosciences. His work aims to connect what we can measure about an animal's basic survival skills and capabilities with what we can measure about their environments and how they can change through space and time. This allows him to predict both the past and the present and future distribution of different species. He works on a wide range of animals, but with special emphasis on insects and reptiles. And today, it's all about the grasshopper. I'm going to introduce Michael with a little bit of a song. This is the final song. Might be a little bit more niche, but pop into the chat if you can recognize it. I don't need sex to reproduce. I can use pathogenesis to clone myself. I don't need sex, and I'll never go back because I'm just as ecologically successful. All right, I'm going to pass over to Michael now, but don't forget that you can send questions into the chat as we go, as they pop into your head. And Michael's also going to be asking questions of you as we go, so pop your answers in the chat too. All right, over to Michael.
Thanks, Kat. Hi, everybody. Thanks for coming along. I'm also on Wurundjeri land, despite the appearances. It's actually Warlpiri land behind me in the Australian desert, and the Australian desert will be important in this talk, as you'll see later on. Share my screen so we can all see the slides now. Just get myself organised here. Sorry, I'm just removing a few things that are, all right, here we go. So yeah, we're talking about sex and, or the lack of. It's a fun topic to talk about, but it's also a really deep and mysterious evolutionary problem. And the star of our talk today is a grasshopper. You can see it pictured here. It's a kind of matchstick grasshopper. There are about 250 species of matchstick grasshopper in Australia. It's a unique group. You'd only find them in Australia. They've got no wings. They look like a matchstick with legs. And these humble little creatures have got a lot to tell us about evolution. And what I'm talking about is summarized in a recent paper that we had published this year in the journal Science. And you can see all the co-authors of that paper in my title slide. So it was a big team behind this work that I'm telling you about. And I'll introduce you to some of the people in that list as we go. So first of all, what is sex exactly? There are a whole lot of different meanings to the word sex. One of them, of course, is gender. So you can see in this particular photograph here of matchstick grasshoppers, we have a male and a female grasshopper. So they're the biological genders. A lot of males around. I'm one of them. Biological males, roughly 50% of the population of a given species. Here's my first question for you. What's the point of males? What use are they? What uses can you think of for males? Put them in the chat, see what you can come up with. So that's one aspect of sex, the gender. But there's another aspect of sex, which is the actual act of sex. You can see a copulation is going on between these two grasshoppers. And what's going on there, of course, is the process of fertilization. So it's a way of getting the sperm and the eggs together. And that is getting us towards the genetic meaning of sex. And that's really what I'm wanting to talk about here today, is the genetic meaning of sex. So what's happening is you've got a set of genes from one individual merging with a set of genes from another. So you end up with twice as many genes. That's called the diploid phase. And what happens then, if you remember your high school biology, is that the diploid phase is producing sex cells that have half the number of chromosomes. And that's through the process of something we call meiosis. So you have inherited 23 chromosomes from your mum and 23 chromosomes from your dad, from your biological parents. And so that makes you a diploid individual. And if you were to make sex cells, eggs or sperm, you do this through meiosis, which is essentially shuffling those cards. It's like you've got a set of cards that have 23 types of cards, and you've got 2 copies of each type of card. And the meiotic process is dealing out those cards, one of each type, to what ends up being 4 different cells. Each cell gets a random one, either your mum's copy of chromosome 1 or your dad's copy of chromosome 1. And not only that, those cards, it's like you chop some of them up, and you stick the cards together from the mother and the father. So you mix them up by chopping up the cards and sticking them together. So basically there's a shuffling of the genes in 2 different ways, so that the eggs that are produced, or the sperm that are produced, are a different combination of the genes that you got from your mother or your father. And then of course the fertilization process brings them back together. So they've halved and they've come back together into the full number. So you go from 23 chromosomes in your eggs and sperm to back to 46, which is the normal number you have in the cells in your body. So that is quite a complicated process. And even though it seems so normal to think of males and females and sexual reproduction, and it's such a big part of our lives, it would be far simpler to get rid of all that complexity, get rid of meiosis, get rid of fertilization, and get rid of males. Then you would simply be reproducing by mitosis, effectively by mitosis, which is the way that your cells are being produced in your body right now. When you grow, you make a new cell. It's just a cloned version of one of your cells. This would be a much easier and simpler way of life. And the thing is that it hardly ever happens this way. And this is actually a paradox. So another question for you just to think about is, what would be the advantages of sex? So see if you can think of what the advantages of sex might be and put them into the chat. But while you're doing that, I'm going to tell you about the problems of sex, because this genetic thing of sexual reproduction produces a very big evolutionary problem. So what we expect in evolution is the things that leave the most descendants, the genes that leave the most descendants, are the ones that we see today. The ones that have more offspring are the ones that are going to be most successful and going to dominate what we see today. And the paradox of sex is that it has a massive cost. And the cost is because of the fact that males are produced. So a sexual female is producing half of her offspring as male and half of her offspring as female on average, whereas a parthenogen is only producing females. This is a very simple concept that took a while to get for evolutionary biologists to come up with, but they realized that actually the sexual female is only producing half as many babies as the parthenogenetic female, because all of the parthenogen's offspring are female, all of them can have babies. So that straight away makes a twofold cost, or imposes a twofold cost on sexual reproduction. So sex is costly from a genetic point of view, from a natural selection point of view, and that's a big mystery. And it's not only costly just because of the twofold cost of having half of your offspring kind of useless males in the context of being able to physically have babies, it's actually a bit of a risky business and a time-consuming business finding a mate and courting a mate. Also, there's a risk of sexually transmitted diseases because you have to get together to bring your genes together, and that creates the opportunity for parasites to be transmitted. So that's a problem. And also, if you have a good combination of genes going, sex shuffles them up, breaks them up. So these are all problems. And for this reason, we would expect to see parthenogenesis everywhere, but it's not. So for instance, there are over 5,000 species of mammal. Not a single one is naturally parthenogenetic. 10,000 birds, none are parthenogenetic. There are 8,000 species of frog and 34,000 species of fish. None of them have managed to get rid of males and reproduce without sex. Lizards have done a pretty good job. About 40 species out of the 6,000 species of lizard can reproduce without males and without sex. And in other groups, like the stick insects, it's actually quite common. And there are some groups that are only reproducing without sex. Overall, about 1 in 1,000 species reproduce without sex. So it's a really low number, and it's really puzzling. But one of them is this Australian grasshopper, Warramaba virgo. There are 11,000 species of grasshopper in the world, and only 2 of them have managed to evolve to be parthenogenetic, and both of them live in Australia. There's this grasshopper and another very closely related one. But we're going to talk about Warramaba virgo. This is a grasshopper that's been known to science as a parthenogen since 1962. It was discovered by a person called Michael James Denham White, who you can see pictured here, and he was a biologist, a geneticist who was at the University of Melbourne. In fact, he started the genetics department after being chair of the zoology department. And he was studying these little matchstick grasshoppers because he wanted to look at their chromosomes and study evolution that way. That's how people studied genetics in the old days, in the 50s and 60s when he was working. He was catching grasshoppers, he was getting males, he was getting their testes out, squishing them on a microscope slide and looking at their testes and trying to look at their chromosomes that way and understand evolution. So he was traveling around Australia, catching these little grasshoppers, often with his family, and he went out to a place near Hillston in Western New South Wales where that green dot is, over here. And there they were searching on these acacia bushes called Acacia wilhelmiana. And in fact, his son was helping out. This is his son, Nicholas White, grown up now, but he was, I think, about 10 or something at the time. And he said, "Dad, I can only find females of this species." And Michael was busy squashing grasshopper testicles at the time and said, "Go away, find more, you'll find males eventually, keep looking." And he came back with 100 females and said, "Dad, there's no males." So actually Nicholas was the discoverer of this extremely rare and interesting case of all-female reproduction in the grasshopper. Michael White initially thought that parthenogens were doomed because they don't have any genetic variation. And so he described it as this grasshopper living precariously on one type of acacia in this small part of the world, and was doomed. But then he found it was everywhere in New South Wales, in inland New South Wales, on quite a few different species of plant, not just the one acacia that they first found it on. So he was still maintaining, though, they were still doomed and not very successful. But then he drove all the way across to near Kalgoorlie in Western Australia and found them all the way over there. And in his field notebook, he wrote, "How wrong can one be?" And so he set about trying to understand how did this species evolve. And over in Western Australia, he and there's this big gap you'll see though between the two populations. So this is includes a feature called the Nullarbor Plain. So you may have heard of the Nullarbor Plain. This is my third question for you. Does anybody know what Nullarbor means? See if you can come up with an answer to that and put it in the chat. I was wrong about what Nullarbor means, but it's actually kind of cool and obvious once you work it out. I'll give it away now. It has something to do with this picture here. This is what the Nullarbor Plain looks like. It's a whole lot of low shrubs, virtually no trees. The Latin for nothing is null. The Latin for tree is arbor, like arboretum. Nullarbor Plain means no trees, and these grasshoppers live on trees, live on bushes, and so that Nullarbor Plain is a pretty big obstacle for them to get across, and so potentially other Flinders ranges. What you'll see on the next slide in green is a species distribution model prediction that we made, where we say, let's look at all the environments where we currently find this grasshopper in terms of climate and soil, and let's look at where else those conditions are similar. And you can see where everywhere in green, it's probably suitable for these grasshoppers to live. You can see there's a whole lot of area here that they're not occurring in that is suitable. And in fact, that's where the other species I mentioned lives. But you can see there's these green patches here along the Nullarbor and also around the Flinders Ranges where it might be suitable for them, or around Port Augusta, but we haven't found them there. But they have managed to travel all this way across the country, even though there are these barriers here. And in fact, we know they traveled from the west to the east because when Michael White was trying to understand how these things evolved, he found another species living in the west, which was called Warramaba P196. I have recently renamed it Warramaba whitei after Michael White, who discovered it. And he also found another species which was called P169, completely confusing, which I've now called Warramaba flavolineata. Now, Michael White originally thought that Warramaba virgo, the parthenogen, evolved by a mutation in Warramaba whitei, to which it looks extremely similar. But one of his colleagues said, I don't think so. If you look at your own results, you'll see that if you stick the genes or the chromosomes of the one in blue there, flavolineata, with the ones in yellow, whitei, you'll find that it's a hybrid. So that's how this thing evolved. These two species, the blue and the yellow dots here, crossed, hybridized, and produced the green species, Warramaba virgo, which was somehow able to become parthenogenetic and get rid of males and get rid of sexual reproduction, and instead clone itself. So we had this army of clones evolve as hybrids in the west and spread all the way across the Nullarbor, all the way to the east. And the sexual forms have not been able to get across to the east. So pretty amazing. What's going on here? The thing that I really wanted to understand was what the role of hybridization was. Lots and lots of parthenogenetic animals, naturally asexual animals, are actually hybrids. And I wanted to know why. Now, do any of you can any of you think of a hybrid animal or plant? If you know of one, type it into the chat. There's some that are very familiar to us. Maybe some of you put this into the chat, a mule. So a mule is a hybrid between a horse and a donkey. Horses and donkeys separated a few million years ago, but if you stick their genes together, you can make this thing called a mule, which actually is a bit stronger, a bit more robust, has a bit more endurance than a horse or a donkey. It can't reproduce, it's sterile, but it is better in some ways. It has vigor, hybrid vigor. So that was one idea I was wondering about. When these two sexual species crossed, did they make something that had hybrid vigor? The other possibility, though, is that they could make some kind of malfunctioning monster, because you have had these genes separate for, in the case of these grasshoppers, we know it's about 3 or 4 million years. Stick them together and expecting them to cooperate well may not work out so well. And it may be that they're actually suffering these parthenogens. And that might be why they're having, they're able to, like, normally sexual reproduction is going to dominate, but this parthenogen, maybe it's having, I guess this would help us understand better what the limitations are if we find out that actually these things are suffering. And so maybe their twofold reproductive advantage is helping them get around the suffering, but that's it. So it could be that they're getting benefits from being a hybrid. It could be that they're actually suffering from being a hybrid. The other thing that could happen through a hybrid origin is you could have lots of hybridization events between lots of different individuals of those sexual species and end up with a diverse army of clones. And that's the thing I was really curious about. Did we have a whole lot of clones living out there in the desert or not? So we went out into the desert and started trying to work this out. This is back in 2004. My wife and I started collecting these animals. We caught a lot of animals from all over the place. And then we brought them back to the lab and measured a whole lot of things about them, about their genetics, and also about their physiology and life history and that sort of thing. So you can see we've got Vanessa and Moshi and Mark Blackett and Ari Hoffman were helping us with the genetics. Jacinta Kong, a PhD student, Ian Atkinhead and Stephen Chown, physiologists. We measured a whole lot of things. So we did the genetics from all around their range. We got populations of the parthenogens and the sexuals, and we compared them. So all these different populations you can see in the colored dots from the parthenogens and all the crosses here, or the pluses here from the sexuals. And this is a plot of the genetic diversity. So this is the blue one, the sexual one, lots of diversity. This is diversity on the y-axis, lots of diversity in the other sexual species, but nothing in the parthenogens, hardly any. This is a plot of how genetically similar they are. And you can see you've got the two sexual species in blue and yellow and the parthenogen in the middle. They are definitely a hybrid, so that's for sure. But the diversity is extremely low. There's a little bit of population difference within the sexuals, like all of those points there, they're so similar, but if you zoom in on it, you'll see that the populations group according to where they've come from. And these are the Western Australian ones, and they're not that different to the Eastern ones. So this tells us that they're actually relatively recent. But they're probably, by recent, we mean about 250,000 years. So actually that's a pretty long time. They've been doing this for 250,000 years. But genetically, there's just one clone. This is like a barcode representation of their genotypes. And you can see a whole lot of variation in the top one and the bottom one, which are the two sexual species, but it's just one barcode essentially in the parthenogens. There's hardly any variation. So that really nailed the answer. Are they a diverse set of clones? There's a typo there, that should be an E there on diverse. No, there's just one. So that huge geographic range there is just one clone. That's all the different sorts of environments that one clone is able to live in. And so then we measured a whole lot of things about them, about their heat tolerance, their cold tolerance, water use, energy use, temperature responses, body size, egg size, hatchling size, clutch weight, total offspring, time to hatch, time to mature, how long they live. You can see the plots on the left-hand side. We measured thousands of grasshoppers to see, were we seeing any of the evidence of them being more vigorous or hybrid monsters? And essentially, no, most of the time there was not that much difference. Or if there was, maybe this is cold tolerance, where you can see this is how heat tolerance. This is how heat tolerant the grasshoppers are. And whitey eye on the left had the lowest heat tolerance. Flavolineata on the right had the greatest, and the parthenogens were just in the middle. That's exactly what you'd expect with hybrids, all else being equal. And you can see these are the faces of these cool grasshoppers, and you can see that the Flavolineata has a stripe on its face and in the middle, and the whitey eye doesn't, and the parthenogen is some combination. So generally they're just a combination. They're not anything particularly special. So we can eliminate hybrid vigor and hybrid breakdown. So we're still a little bit puzzled. What this seems to suggest is that it's simply hard to evolve. It's just a really hard thing to evolve. When it does evolve, it's successful. We tried to make these parthenogens by crossing them in the lab, and so did Michael White back in the 70s. And you can see one that Michael White made. He made one that seemed to possibly be parthenogenetic, and these are a number of individuals we made that were hybrids, but they weren't able to lay many eggs, so we didn't make any parthenogens. We're hoping to do this on a larger scale and actually successfully make a parthenogen, but so far we haven't had much success. And you remember I said that there aren't that many species of pathogens around, and in fact the mammals have an inherent problem with becoming parthenogenetic. That's actually Dolly the sheep, and you may remember that she was a clone, but you actually can't make a parthenogenetic mammal by putting a sperm and an egg together. You actually have to take the genetic material out of one mammal cell and put it into another cell that has been through the body of a male. That's called genomic imprinting. So there's an inbuilt mechanism to stop parthenogenesis in mammals. In birds, you occasionally get parthenogens, in that you can get parthenogenetic turkeys, but they only have male offspring. So that's the end of that. There are some frogs and fish that can clone themselves, but they need sperm to trigger their eggs, so they can't break free of males completely. So there are all these limitations on things becoming parthenogenetic, and we're thinking that maybe that's the simple reason why we don't see them very often. But where we do see them a lot, in one particular place, the scene behind me in the Australian desert, we've got the grasshopper, as you know. There's a stick insect as well called Cypoloidea. They feed on bushes of the mulga tree and also on cassia bushes, and it turns out those two kinds of plants are also asexual, parthenogenetic. And as you know, there are lizards that are parthenogenetic, and one of those is the spinose gecko, which lives under those bushes. running around at night. And during the day, there's another lizard in the Australian desert called grey skink, which is also parthenogenetic. The grasshoppers eat the plants, the lizards eat the grasshoppers. And so we have a whole food chain of parthenogenetic animals living out there in the Australian desert for some reason. So that's really most of what I've got to say. We're really still puzzled about what's going on with parthenogenesis, but we're pretty excited that the biodiversity that we have in Australia is helping us understand this problem, especially the fact that we've got so many natural pathogens in our deserts. And so we hope to use this amazing biodiversity to help us understand more about life on Earth. And we hope that we can manage to preserve and continue to preserve most of that biodiversity. It's so important for so many reasons, but especially in this case, understanding the basic fundamental questions of life. So thanks very much for your attention, and I'm very happy to answer any questions you have.
Oh, thank you so much, Michael. Clap, clap, clap from me. I can imagine we're all clapping, but I'm going to clap on everyone's behalf. I'll just get you to stop sharing your slide.
Yes, just process that.
Everyone can see your face. Everyone feel free to pop questions into the chat and I'll pass them along to Michael, but I might start off with a few of my own. You said that it's really hard to evolve pathogens, but what about all of this endocrine disruptor waste that's going into our waterways and things like that? Do you think that that kind of thing might impact the evolution?
Those sorts of pollutants are affecting the hormones. So they're not affecting the genetic system. What's happening in the evolution of parthenogenesis is the actual genetic system is being modified.
Yeah.
And actually what's happened in both that gecko, the Binos gecko, and the grasshopper as to exactly how they've changed their genetic mechanism. They actually just double their chromosomes before they do a normal meiosis. It's that sort of simple change. That sort of thing is unlikely to be triggered by those endocrine disruptors. Yeah, so I think that's not necessarily going to cause parthenogenesis. It certainly affects this, the expression of sexual characteristics, though.
Yeah. So I wouldn't necessarily think that they're directly sort of linked, but I wonder because some fish species are becoming feminized. Is this potentially evolution's out of like being able to keep going? But
Oh, good question. Yeah.
So your next research question. I also wonder, in terms of them being successful, you sort of, clearly show that parthenogens can be successful, but it sort of makes me think about inbreeding. And Gayatri points out that, in cases like inbreeding, you don't eliminate bad traits. So like, what if, there's this bad trait that then's just being passed on?
Yeah, so these grasshoppers are not inbreeding; they're cloning. So inbreeding, you sort of some pathogens actually do have an inbreeding mechanism of parthenogenesis where they produce their egg cells and then they let those egg cells fuse to each other, and that is essentially like selfing or inbreeding, and that causes problems because it exposes the bad copies of your genes. Everyone's got some bad copies, but they're usually covered up by a good copy. But the problem for parthenogens in the long term is that there's a certain number of mutations that the parthenogenetic population has of bad mutations. And what a sexual species can do is they can stick two bad copies together into one individual and lose that from the gene pool. Whereas the parthenogens, their mutation load just ratchets up. That's the metaphor. Like it only gets worse. So there's over time, you might expect that they might end up in mutational meltdown because they just accumulate so many mutations. So that's more the problem for them. It's not so much inbreeding, it's getting lots and lots of mutations over time and not being able to get rid of them.
Yeah. Thank you. Kieran has an interesting question. Can we expect these parthenogenetic females to have reproductive hormones, or would it be slightly different to maybe a sexually reproducing one?
That's a great question. They really evolved, just moments ago from an evolutionary perspective. So they actually still mate. They have all the normal hormones, all the normal responses. If you put them with a male, they'll quite happily mate with that male. What do you think would happen though if a parthenogen mated? What would happen to her eggs? Does anyone know? What would that mean? Because the parthenogens not doing meiosis. So she's producing eggs that have 2 sets of chromosomes.
So you'd get like, well, I'm thinking of trisomy, but that's only like one double. You'd get like a whole
Triploidy is the
set.
word.
Yeah,
It's
triploid,
you
that's
ended
it.
up with 3 sets of chromosomes. And we can make this happen. We can mate the males of either of those 2 sexual species with the parthenogens, and they produce babies that have 3 sets of chromosomes. We never see those in the wild. But so yeah, so they're actually just, you know, in almost every way, just normal, except for the way they make their eggs.
Mm, mm. Pressures of testing my genetics. Alina asks, might the asexual reproduction be an advantage in something like a desert environment if it's harder to find a mate?
Yes. I mean, I guess I was always thinking, oh, there was a bit of evidence that there were lots of clones, and there was, you know, there's a lot of, you know, species where there's hybrid vigor, and I sort of was sold on for various reasons, on the idea that it was the hybridization was really what was making them successful. But this study we did has just completely destroyed that argument. And so it made me go, okay, yeah, so, but that is a huge advantage if you don't have to find a mate in the desert. And, you know, you can start a population from one individual. So there's no inbreeding problems like we were discussing before. There's no inbreeding. You can start one individual and build up a population with twice the normal population growth rate. And you can also think of that twofold reproductive advantage as, you can think of it as their population growth rate's twice as high, but you can also think of it as them being able to tolerate twice the mortality that a sexual species can have. So yeah, it's really powerful. And it's probably the reason why they were able to manage to get across the nullable plane. And their sexual relatives didn't. So this could be something to do with why we see parthenogens in the desert. Good question.
So in terms of the species where you might find this, are you more likely to find them in more like animals that are like living in solidarity? Solitude yeah. Is the word I was looking for.
Yeah, you can think of it as solidarity too. They're all like solidarity in cloning. But yeah, the broad pattern across the world is for parthenogens to be in places where the glaciers were. So about 20,000, 18,000 years ago, a lot of the world was covered in ice, and where that ice was, it's retreated and things have moved back in. And if you look in the Northern Hemisphere, you'll find parthenogens are there, and the sexual species that they evolved from are on the mountaintops that were never under the ice. So that's a really big pattern in Europe and North America. In Australia, it's in the desert, but similarly, our deserts would have become much less inhabitable during the glacial times because it was a lot drier, and a lot of the sand dunes, you know, like behind me, would have been active. And so I think it's possibly quite similar. You've had these areas that were uninhabitable during the glacial times that the parthenogens have been able to invade and find a place to live. So yeah, that could be something to do with it as well.
And that sort of leads really nicely into another question from Kieran about whether the switch between whether you're doing asexual or sexual reproduction, is that dependent on availability of food and resources?
Yeah, it is. So some species can do both. They can switch between. So, you know, some water fleas, they'll reproduce asexually until their pond dries up, and then they'll produce sexual eggs. And that allows them to have a diverse range for the next pond when it fills up again, which might have a completely different range of other animals and plants living in it. So that's actually really interesting that some species can do both. That someone long ago came up with this idea called the balance hypothesis, which was really saying there must be an advantage to sexual reproduction, because the species that can do both, if there was no advantage, they'd just stop. They can do both. Why do they keep sex? So there's some advantage. And it does potentially have something to do with changed environments.
I also wonder if it's sort of like, you know, we have these vestigial organs and things like that, but I guess this is like a thing that's across the animal kingdom, not just one animal.
Yeah.
Alina asks, can the triploid grasshoppers, so, you know, if you do have a triploid grasshopper, can they survive normally, or would they be negative?
Yeah, that's a really good question. And yes, they survive. They grow up and they have babies, and you can get babies from their offspring, from them. So they actually they seem fine. But I have been looking through my data on these things, and I think what's happening is that they take a lot longer to mature. So I'm not, this is sort of a little bit sort of preliminary, but there is something different about them, and it may mean that doesn't work to, you know, fit your life cycle into 1 year if it takes a lot longer to mature. But just watch this space.
Everyone stay tuned.
Yeah.
Kelsey asks how you'd expect the hybrids to persist in changing environments. And I think right now, the environment very much is changing quite rapidly. And, you know, given that they may have less genetic variation for selection to act on, how might that change?
Yeah, I know. It's fascinating that they've managed to, I find it amazing that they're able to live across such a diverse range of environments, that one clone too. That just shows you one individual, one genotype has just got a lot of capacity to live in a lot of different places. It's a very versatile genotype. That one genotype can feed on about, I don't know, 3 different genera of plant and probably about 20 different species. It's living in all sorts of different predation environments across its range just now in space. And then you think about the changes in time. And if we're right that this thing has been around for about 250,000 years, they've gone through a whole glacial cycle where, it got really cold and dry, and their range would have contracted to quite a different place. And then they've probably had a whole different range of other species living with them during that time. So, it does tell us something about how robust even one genotype can be, that they've managed to survive through all that change. It really does make you think, question your ideas about the role of genetic diversity. But I could add though, there's one really big important hypothesis about the advantage of sex, and that's in relation to the changing environments caused by pathogens, parasites. I don't know if you remember the picture of the lizard that I showed. It had a whole lot of red dots around its eye, and they were parasitic mites. And actually those parthenogenetic versions of that lizard are 150 times more likely to have those mites. One of the advantages of sex is potentially basically changing the locks of the immune, with the immune system rapidly by mixing the genes together to cope with pathogens that can evolve fast. And maybe if we were clones, if we just had one woman was our clone was our population in the world, then maybe COVID would have knocked us out.
Oh. That's a really interesting thought. And like, as an immunologist myself, I'm just like, oh, let's latch onto this. So do they potentially have weaker immune systems? Because I know at least in humans, part of what attracts us to a mate is people with different MHC, like, so,
yes.
Different immune systems to us. So like, how does that impact your immune system?
Yeah, we do find the grasshoppers sometimes have this parasitic fly. It's a fly that lays its egg on the grasshopper, and then the little larva goes into the grasshopper and eats it on the inside, and then this pupa pops out. The grasshopper's still alive. It's kind of gross. But, they seem to infect the sexual species just as much as the parthenogens. There's no disease we've noticed that these things have. It may be that it's just not a great place for disease transmission in the drier desert environments, and that might be part of the reason why they can cope well. Hey, one other really interesting pattern too is that there is no case of parthenogenesis in the sea, not a single one.
That's interesting.
Whether that has something to do with disease transmission through water, I don't know.
Yeah. Different air versus water. You mentioned that there are 250 species of matchstick grasshoppers in Australia, and that they're only in Australia. Is there a particular reason that they thrive and survive here and not elsewhere?
Well, it's sort of similar to why we only have monotremes in Australia. Like, it just so happened that a group that evolved into the matchstick grasshoppers came into Australia and then diversified, and they haven't been able to leave Australia. So it's really a historical explanation. And there are other similar sorts of grasshoppers that are wingless that are distantly related, but this particular group made it into Australia and then went kind of crazy and diversified. So it's really an accident of history in many ways.
Kieran asked, maybe if you wanted to comment on reproduction that's mediated by organisms like Wolbachia.
Yeah, Wolbachia is a bacteria that lives in the cells of insects, and it wants to transmit itself by being passed on through the female line. And so Wolbachia causes the species that it infects to change in ways that promote the females transmitting the Wolbachia in the eggs. And one way they can do that is to cause parthenogenesis to occur. That is not what's happening in these grasshoppers, but in other species, yes, Wolbachia is actually causing parthenogenesis to occur somehow. And if you were to cure the species of the Wolbachia, then it would switch back to sexual reproduction. So good point.
Yeah, and I think there are a lot of great scientists here at University of Melbourne who are looking at Wolbachia and its use in mosquitoes.
That's right. That's my colleague Ari Hoffman, and Ari was pictured in he's one of the people working on this project with me. Yeah.
Little shout out there.
Small world.
Yeah.
Yeah.
So Nigel asks, as in bees, sexual reproduction and parthenogenesis, it leads to different sexes among the offspring. Is it the same for grasshopper? I know that you sort of showed your tree of it all sort of being female.
Yeah, it isn't like that in the grasshoppers. So they actually, the sexual species, one of them has an XY sex determining mechanism, and the other one has this thing called an X1, X2Y sex determining mechanism. It's a bit quite complicated. The parthenogens, though they're just genetically female, they have two X chromosomes, and so they just clone those two X chromosomes, and you don't ever get males.
So what's next in terms of your research? You've gone to collect lots of grasshoppers, which kind of looks like a fun journey, but where to from here? Do you look at new species, or do you sort of see where this takes you?
Sorry, Kat, my computer just was playing up for a second. You were asking where we're going next with this.
Yeah.
Yeah, so one thing we'd like to do is further understand how diversity might evolve in these species, in these parthenogenetic species. We want to see what happens to the genome when you make these hybrids. Does some genes get turned off? Because you're sticking these two genomes together. Somehow they're working. It's potential that there's some interesting genomic effects of becoming a hybrid. So we want to look into that a bit more. We'd really like to understand why hybridization causes the changes in meiosis that allow them to clone as well. That's not really clear at all either. And to do that, we'd really like to try and make our own parthenogens. So we like I mentioned in the talk, we have tried on a small scale, but we're much better at keeping these things in the lab now. So I'd like to make a whole lot of crosses and just see if we can generate our own parthenogens, and then we have something really powerful to compare with the natural ones. So there's some of the things we'd like to do in the future.
So to that end, can I ask, how many grasshoppers do you have in the lab?
Right now, we don't actually have any of those ones at the moment. We're on pause with our work on that species, but we do have a few hundred in the lab at the moment. There's another project with the matchstick grasshoppers we're working on where we're actually, I mentioned the wingless, and some species are actually of conservation concern because they're being lost from habitats, and we're needing to give them a helping hand and translocate them to good habitats. So we've actually just been translocating thousands of them at the moment. So we've got quite a few in the lab, but they're sort of coming in and then going out. You might have heard about some of this happening in Royal Park. Actually, we just translocated some into Royal Park, about 1,000 of them.
Nice and close.
But that's another story.
Well, we've run out of time, but this has been really, really fascinating. So thank you so much to Professor Michael Carney for an amazing presentation on, I guess, the costs of sex and the benefits of sex in the animal kingdom, and how hybrids are not monsters, but can do pretty okay in the ecosystem. So thank you very much, Michael. And thank you, everyone. Thank you, everyone, as Michael just said, like for your questions and for your participation and your comments, your answers to questions as well. You can read more about Michael's work with the links that are in the chat, the paper that was published, as well as some other articles about the work. And I also just want to say this is the end of the masterclass series, and I want to thank the people behind the scenes. This isn't in the running notes. They don't know I'm doing this, but I wanted to thank Amber, Alexa, Ben, Daryl, and all the other Faculty of Science and Faculty of Veterinary and Agricultural Sciences staff who are behind the scenes. But this isn't the end of Science Festival. There is lots more happening today and over the weekend, so check out the schedule, and thank you everyone for tuning into the masterclasses. Have a lovely afternoon. Thanks, Michael, and thanks everyone. Bye.